Okay. I think I should be able to get confirmation from someone in the crowd that we're live, or from my team, but I'm pretty sure we're live now. So welcome everyone to session 6 of Neural Launchpad. We're really excited. We have a completely different topic array today than what we have the couple last days. So I won't be talking for long, but just so everyone's aware, this is session 6 of 10, and we're excited to have 4 more weeks after this. I'm gonna post something onto the chat right now that if you want to hear anything from us once a week, just one single email, we won't spam your inbox or anything like that, with the talks for that week, just once with an announcement, just feel free to register for that little listserv, and that's all we're going to use it for. But it kind of just helps us keep tabs on like who's attending and have a more easy way to contact everyone. So with that being said, it's my pleasure to introduce our 2 moderators for this session, which is Dr. Takaki Komiyama, who is from UCSD, and Britton. Britton, I didn't ask you how the correct pronunciation of your last name is, but I will say Britton's here, and he is at HHMI and Janelia's campus. And so we're really excited to have them 2 here to moderate. And with that, I'm just going to hand it off to Dr. Komiyama to introduce our first speaker.
All right, thank you. Yeah, thanks for letting me be part of this. It's a very exciting thing you guys are doing. All right, so the first speaker is Joe Zach, who is in Benke Murphy's lab at Harvard, who will tell us about nonlinear stimulus encoding in the olfactory system. So please start.
All right, let me get my video going here. All right, can everybody see my screen all right? All right. So first I want to extend my thanks to the seminar organizers for the opportunity to tell you about my work. I'm really excited to be here virtually, and I'm looking forward to the discussion to come. So one of the broad questions that I'm interested in is how blends of sensory stimuli are recognized, detected, and encoded by the brain. And to answer these questions, I use the olfactory system as a model. And the basic biological problem that I'm interested in is this. Despite a relatively diverse and complex array of receptor types in the olfactory system, humans have a few hundred and mice over 1,000, the number of possible odor molecules is virtually limitless. Now, because each molecule activates a subset or a fraction of the receptor types, even relatively simple mixtures of odor molecules may saturate the entire complement of sensory receptor types. And this is really a fundamental problem for stimulus encoding. So the question is, how does the olfactory system solve this problem? And before I begin, I want to start with the relevant anatomy of the olfactory system as it pertains to my talk. So in mammals, olfactory sensory neurons are found in the olfactory epithelium, and these neurons are sensitive to a molecular feature of an odor molecule, although they can also bind different features across a spectrum of affinities. Now, throughout this talk, I'll refer to these neurons as either sensory neurons or OSNs for short. Now, sensory neurons of a common type, meaning those that express the same OR, project to a well-defined structure on the surface of the olfactory bulb called a glomerulus, where an initial round of information processing takes place. And from there, information is then routed to higher order structures in the brain, including but not limited to the canonical olfactory cortex, also known as the piriform cortex. So coming back to the problem that I framed earlier, in this cartoon here, the large circle represents the olfactory bulb, and the small circles within it represent individual glomeruli. Now, when this animal encounters an isolated odor, a sparse pattern of these glomerular inputs becomes activated. And we can think of this as somewhat analogous to this scene here, where we have this very old building sitting against a void of a background. Now, if you've ever spent time in downtown Boston, you'd recognize that this is the old State House. And if you know that this is the old State House, you'd also know that this building doesn't sit against a void of a background, but rather this somewhat chaotic scene of more modern buildings. And much like the old State House doesn't exist in isolation, neither do odors. And we can think of these as background odors, which at the same time increase the number of active glomeruli on the surface of the olfactory bulb. However, if we add too many odors to the background, we raise the possibility of activating nearly all the glomerular input channels. And this is similar to saturating your visual scene on the right, where we can no longer make out the old State House at all against the background of the buildings that surround it. So the problem becomes, how does the olfactory system prevent input layer saturation against these complex backgrounds? And one potential solution to this problem may arise through a neural computation called normalization, which readjusts the activity profile of sensory inputs to maximize their collective dynamic range. And I'm going to tell you about a mechanism derived from the natural statistics of receptor ligand binding in the olfactory system that could help to facilitate this computation. But I also want to mention that this is a shared problem across biological sensing systems. Many signaling pathways share a convergent architecture with ligands interacting with receptors in a many-to-many fashion. As we can see in the simplified illustration here of a generic signaling pathway, many ligands compete for the same receptors, which then each activate or suppress the activity of an effector molecule, which then ultimately acts at a target gene. Now, to be more concrete, the importance of filtering a complex background is shared by both the olfactory and the adaptive immune systems. In the latter, lymphocytes must quickly and accurately identify a fraction of foreign ligands in a sea of native ligands. And it's competition between native ligands as well as inhibition between native and foreign ligands, which determines whether an immune response will be initiated or not. Now, many of the same principles that I'll describe are also at play in other systems, both neural and otherwise. Therefore, this is an important problem for understanding how a limited set of receptors in any biological system could interact with limitless varieties of ligands without becoming completely saturated and thus ineffective. Now, since I'm going to be talking about receptor ligand binding statistics in relation to olfactory sensory neuron activity, I want to briefly describe how these sensory neurons are activated by odor molecules. Now, the olfactory transduction machinery is found on the cilia of sensory neurons, where odor molecules bind to olfactory receptors, which are coupled to a G protein, which sets in motion the somewhat complex downstream signaling cascade that ultimately results in a pattern of action potentials being sent to the brain. Now, as I mentioned before, multiple odors can in fact be bound by the same receptor type, but they engage the downstream pathway with different efficacies. In this example here, the blue odor generates less spike, despite the fact that it's interacting with all the same molecular components. Now, our question is, what happens if both of these odors are present at the same time? Is the output from the sensory neuron simply a summation of the activity transduced by either of the two odors alone, or is it more complex? Now, importantly, much of the prior work in this area makes the assumption that a firing rate of a sensory neuron is set solely by the binding affinity of the receptor complex for ligands, and that mixture interactions arise through what's called syntopic or competitive antagonism. However, in many cases, a single free parameter, which is given by a model of competitive antagonism, does not account for OSN mixture responses that are observed in existing data sets. So in the next few slides, I'll describe our attempts to develop a new model that more faithfully captures stimulus mixture dynamics in olfactory sensory neurons. And given the shortcomings of the syntopic model or competitive antagonism, with my collaborator Gautam Reddy, we developed a new model of how odors may interact with olfactory sensory neurons. In our model, given by the equation here, the response of a sensory neuron is defined by its maximum physiologically bounded firing rate and two other free parameters. We call it kappa and eta. And here, n is the Hill coefficient. In our model, kappa defines the affinity of the odor receptor complex for a given ligand or its binding sensitivity. And eta defines the efficacy by which the receptor complex activates the downstream signaling cascade. Now, for example, it might be possible to have an odor that binds to the receptor complex very tightly, but only weakly sets in motion the downstream signaling cascade. And I should mention that this idea is not necessarily unique to olfactory signaling, but we took inspiration from other G protein coupled systems. Now, different odors will each have their own kappas and etas, and we can model mixture responses of multiple odors by simply just adding together some terms in our model. And to simplify this a bit more, if we have the two response curves for two pure odors across a range of concentrations, we can extract the kappa and the eta from each. So for odor A, kappa is the point where the rising phase of the sigmoid begins, and eta is the asymptote. And the same is true for odor B. And with these terms, we can plug them into our model and make a prediction of where a mixture of these two odors at any concentration might fall. And importantly, our model is able to capture all types of nonlinear mixture interactions that are observed in existing data sets, including both synergy as well as suppression. And in fact, our two-step model made some other interesting predictions. If we plot here the fraction of active glomeruli as a function of an odor, the complexity of an odor mixture on the x-axis, the rise of this curve here is set by this term which we call rho, which we define as the antagonistic factor. And rho is determined by the correlation between the log inverse of all of the kappas and the log etas of a population of olfactory sensory neurons. Now remember, these are the two terms which determine the response curve shape for an individual pure odor. And it turns out if these two terms are well correlated across a population, what we see is that the number of active glomeruli rises very quickly, and the entire complement on the surface of the olfactory bulb becomes saturated for even very simple mixtures, less than 20 different components. So here we assume that each odor provides input to a constant fraction of glomeruli with some overlap between them. However, if there's even a modest decorrelation of these two terms, what we find is that the response curves rise much more slowly, thereby leaving inactive glomeruli for high complexity mixtures and desaturating olfactory input. So another way to depict this data is the cartoon at the right here. Each small circle represents a glomerulus, and the dark circles represent active glomeruli. For a single odor, we see that a fraction of these glomeruli are active. However, if kappa and eta are well correlated, what we see is that the number of active glomeruli rises very quickly and then saturates the entire complement by 16 odors in a blend. So this is when we have no antagonism. However, if these two terms are completely decorrelated, what we see is that the fraction of active glomeruli remains constant regardless of the mixture complexity. So in short, if there's some decorrelation between the binding sensitivities and the activation efficacies across a population, the activity of sensory inputs to the olfactory system should rise sublinearly with mixture complexity. And the question we hope to address now is how prevalent these antagonistic interactions between odors in a blend actually are in a naturalistic system. And to make these measurements, I made use of OMP GCAMP3 mice, which express GCAMP in all olfactory sensory neurons. And I started by imaging odor responses at sensory neuron axon terminals within glomeruli. Now on the right here is a typical field of view of what these glomeruli look like under a 2-photon microscope. And I'm showing you the calcium responses of two neighboring glomeruli to the odor methylphthalate. We can see that one glomerulus responds quite robustly, while the other not at all. And we didn't just image odor responses at, say, a single concentration, but across a range of concentrations spanning 3 orders of magnitude. And on the bottom, I'm plotting the response curves from 4 different glomeruli, including the 2 depicted on the top. Now, after imaging one odor, I then imaged the response to a second odor, in this case, an odor called isobutylpropionate. And we can see that the example glomeruli now show the opposite response profiles. And we can plot the response curves for the concentration series on the same axes below for the same glomeruli. Now, I then made mixtures of these 2 odors. And we can see from the examples above, plotted in purple now, that the mixture response is below the larger of the most effective of the 2 odors, and that the linear addition of the 2 odor, or the linear addition of the 2 pure odors is larger than the mixture response that we observe. Now we can plot the mixture responses on the same axes below, and what we see is that the mixture responses fall below the response curves for the 2 pure odors, thereby indicating that there's mixture suppression in these 4 example glomeruli. And this tells us that antagonism may potentially be quite prevalent for this odor pair. But how do we go about quantifying this? So I collected a data set of greater than 300 glomeruli from several different animals. And I first compared the linear addition of the top 3 concentrations for each odor against the observed mixture response. So in the scatter plot here, we can see that nearly all the data points fall below the unity line, indicating that the mixture prediction was larger than the mixture observation. Now on the right here, I plot the distribution of p-values obtained from 334 comparisons. And what we see is that in nearly 80% of the glomeruli, we observed mixture responses that were significantly less than what was predicted by a linear encoding model. However, it is possible that the largest responses that we observed for either of the 2 pure odors alone may represent either neuronal firing rate saturation or calcium indicator saturation. So to account for this, I made comparisons between the mixture and the largest of the 2 pure odor responses. And we see that even when being more conservative, we observe significant suppression in roughly 40% of the glomeruli in our data set. However, there's one potential problem to this result, which is that the sensory neurons express some inhibitory GABA B receptors at their axon terminals. In this diagram here, there are laterally projecting GABAergic interneurons that could in principle receive input at one glomerulus and feed forward inhibition onto OSNs at neighboring glomeruli, thereby giving rise to an effect that looks like mixture suppression, but is actually the direct result of neuromodulation of these sensory neuron axon terminals. So rather than taking a pharmacological approach to block the receptors at the axon terminals, I took a manual approach and changed the imaging location to the olfactory epithelium, where there's no evidence for efferent modulation of these sensory neurons at their cell bodies. And to do this, I developed a new thin bone preparation directly above the olfactory epithelium and made sure not to puncture the bone to prevent pressure artifacts in the nasal cavity from affecting the responses we see in freely breathing animals. Now on the right here is an actual image of the sensory neuron cell bodies resting in the olfactory epithelium. And when I play this movie, you'll see subsets of these OSNs respond at different densities and intensities for different odors. On the bottom here, I'm showing you the DF over F responses to 32 concatenated odors from 5 selected OSNs in the field above. And the point that I'd like to get across is that even though that we're imaging these cells through a thin bone preparation, we have pretty excellent signal to noise resolution, and we can reliably extract individual odor responses from these cells. Now, I could then repeat my earlier experiments at the single cell level. So I used the same odor pair as before, and here I'm highlighting 6 selected OSNs and their responses to either of the 2 pure odors and the mixture. And again, what I want to point out is that we can see the purple mixture response falls below the responses of the 2 pure odor responses. And it's generally less than the linear summation of the 2. And I quantified our data in the same way, first comparing the sum of the pure odor responses against the observed mixture response, and also comparing the maximum of either the pure odor component responses against the mixture. And again, I found that antagonism was remarkably prevalent at the single cell level, although it was a little bit less dramatic. We found significant antagonism in 60% of the OSNs using a linear encoding model, and around 30% of the individual cells when using a dominant component model. So I've shown you so far that, I've shown you quite a bit of evidence that these binary mixtures exhibit widespread antagonism at both the population and the single cell level. But if you remember back to the beginning of my talk, one of the key questions that I'm interested in is how is antagonism related to complex odor or mixtures of odors? And what's the relationship to activity patterns of sensory inputs? So the question is, what is the relationship between mixture complexity and antagonistic interactions in the olfactory system? So again, I imaged individual sensory neurons using my thin bone prep, but now in response to complex odor mixtures. So in these experiments, I first imaged OSN responses to 16 individual odors, and I then made blends of odors containing 2, 4, 8, or 12 different components. Now, from the responses of the individual odors, I could then make predictions about what the mixture responses might look like for individual cells. So here, I plot the mixture responses that we observed on the y-axis and the linear prediction for each mixture on the x-axis. Now, the OSN tuning to each of the pure odor components is plotted in bars on the right. Now, to account for the possibility that the largest observed mixture responses sort of here were set by firing rate saturation of the neuron or calcium indicator saturation, I fit each of these data clouds with a sigmoid with an initial slope of 1 and an asymptote in the top quantile of the data. And then I measured deviations from the sigmoid rather than the unity line. Now, I also broke my data down by mixture complexity. And what we can see is that larger complexity mixtures, those that are purple or blue, typically fell further from the sigmoidal fit to the data, and the likelihood increased that they would fall off the sigmoid increased with mixture complexity. Now on the right here are 3 other OSNs in the same field of view. And on the far right is my favorite example here, where we see this OSN that responded to only a single odor alone. But if we made blends that contained this odor plus other non-effective odors, we could completely antagonize the sensory neuron from generating a calcium response, depending on what the mixture blend contained specifically. Now, to quantify our data, I measured the distance from the sigmoid for each mixture, where positive going values represent antagonism and negative going values represent synergy. Now, on the bottom are the distributions for the deviations measured from the 4 sensory neurons on the previous slide for each mixture complexity. And we see that the deviations increase with the complexity of the odor mixture. And of course, it didn't just do this for 4 OSNs, but rather hundreds and hundreds of them. And across the OSNs, we then plotted the deviations from the sigmoidal fit for each mixture complexity as a cumulative density. And what I found was that the magnitude, meaning the size of the deviation, as well as the likelihood of finding a deviation, increased with the complexity of the mixture. And to establish statistical significance from this data set, I used a subsampling approach where I selected several hundred random deviations from each distribution, then compared them to a null distribution with the same standard deviation and a mean of 0. And I repeated this process 100,000 times to build p-value distributions, which I plot on the right for each mixture complexity on a log scale. And across all mixture complexities, each subsample resulted in a significant comparison. Now, lastly, to ensure that our result didn't arise from a few well-responding cells, which could yield the possibility of measuring large distances from the sigmoidal fit, I normalized each cell to the value of the sigmoid and then remeasured the distances. Now, even when normalized, I found that my same basic result held true. Antagonism increases in magnitude and prevalence with mixture complexity. So I've shown you that antagonism is widespread in individual sensory neurons for binary mixtures. The prevalence and magnitude of antagonism increases with the number of odors in a mixture. And then lastly, that antagonism contributes to normalization of input to the olfactory system by suppressing OSN activity resulting from naturalistic-like odor blends. So with that to conclude, I've shown you a mechanism derived through the natural statistics of odor binding of sensory neurons that could desaturate input to the olfactory system and restore sensory acuity despite extremely complex sensory environments. And this mechanism operates through antagonistic interactions between odors competing for the same receptors and their efficacies in activating the downstream transduction cascade. So lastly, I just want to quickly acknowledge a few people. Of course, my advisor, Venki, for guidance on this project, as well as Massimo and my collaborator, Gautam, who drove the development of the modeling work and for their help to conceptually develop the experimental work. I also want to say a quick thank you to my funding from the NIDCD, who has made this work possible through multiple fellowships. And if you have questions, please feel free to reach out to me on Twitter, through my personal website, or via email. Thanks.
Hey, excellent talk, Joe. That was fantastic. And I've been told that I get to ask the first question while Britton curates questions from the chat room. So yeah, please, everyone, feel free to ask your question at the chat. All right. And so, yeah, you showed very convincing evidence that there's a lot of antagonism that happens even at the level of the nose. So that was very exciting. And I believe the idea is this is all happening at the receptor level of single individual neurons. But this observation also reminds me of the observation in Drosophila, where they showed receptor neurons can show ephaptic interactions between neighboring neurons. Do you see any evidence for something like that here? Like, do you see any anti-correlation of neighboring sensory neurons?
I haven't looked for that in the epithelium. These things are, they're not so densely packed at the cell body level, right? They're supporting cells which surround each OSN. So really, to answer that question, we have to sort of look at the level of the axons themselves as they course through the cribriform or as they sort of terminate in the fascicle at the individual glomeruli. And unfortunately, using this approach with the GCAMP3, where we've labeled all of the OSNs, the labeling is too dense to identify individual axons in the bulb. So a different way that we could answer that question is if we could do some sort of viral sparse labeling of axons and hope that we got two that were close to each other and see if there's some sort of coupling between them that way. But yeah, it's a question I would love to answer, but I think we're a little bit technically limited in what we can do given our system right now.
Thank you.
Yeah.
Britton, how are we doing at the chat?
Just to come in. So I've got a couple here. So it seems like downstream circuits should care a lot about discriminability or detection. That is, in a complex odor mixture, can you tell whether the kth odor is present, yes or no, or can you discriminate which odorant has the maximum concentration? So I'm wondering if you tried any sort of decoding analyses on the data to look at this.
Yeah, so we did in the model. We didn't do it on the real data, but we made a simulation where we calculated the mutual information between a glomerular ensemble associated with the target odor. And then we made ensembles of backgrounds that contained different numbers of background odors. So they got more and more complex. And we show that a system with no antagonism really quickly loses information or mutual information between the target ensemble and the background as you increase the background complexity. So by 10 odors in the background, you've completely saturated everything and all the mixture, the information's gone. But if you incorporate at least a little bit of antagonism or a modest amount, you can preserve information longer as the background complexity increases. So yeah, we've done a little bit of that kind of decoding analysis, and we find the optimal level of both antagonism as well as sparsity in the inputs to preserve the most amount of information.
And then on how does that vary with the sensitivity and the efficacy? If you look at the mutual information surface as a function of those two parameters in the model.
Yeah, so we didn't calculate what's we find the value of rho, which is optimal. I can't remember what exactly it is, but it corresponds to with what we would predict from existing data sets. So we could pull the kappas and the ettas out of measurements that people have made in the past with electrophysiological techniques, and sort of found what's the distribution of the predicted kappas and ettas. And what we figure is optimal is really close to what's observed in the data sets that were available to us.
All right. If you guys in the audience have any questions, please feel free to type them into the box here. I wonder also a little bit about the dynamics during natural behavior, because you have this sort of sniffing rhythm. Do you think that the responses of these neurons, and especially to mixtures, is more or less stationary, or do you think that that's somehow modulated by the rhythm?
Yeah, it's a great question, and this is something I'm working on. I'm hoping to extend this work into nonlinear stimulus encoding into both adaptation and antagonism, right? So the sensory neurons adapt at the same time as they're antagonized. So if you have two odor pulses that come close together, the likelihood of a sensory neuron responding at the same level to the second pulse is greatly diminished. Now, if you have competition at the same time, so you're antagonized on the first pulse, it might free you up to be able to respond to the second one. So there's really all kinds of weird nonlinear distortions that could affect what information is getting through, and it's unclear what the importance of the antagonism versus the adaptation is in terms of maximizing the information transfer to the first level circuitry in the bulb.
Very cool.
Kind of a more general question. This might be completely off. Is there any sort of feedback back from the actual brain and like hormonal state and like other kinds of like adrenaline, any other thing that might be influencing the mouse's like overall state that can feed back at the layer of those sensory neurons, or is it pretty much fully just cascading downwards?
Yeah, so there's indirect evidence, or there's direct evidence of indirect modulation of the sensory neurons, right? So they express GABA B receptors, and they also express dopamine D2 receptors. So you can have input from, say, the cortex, which could then synapse with the local neurons in the glomerular layer, which provide that inhibitory drive or inhibitory input onto the sensory neurons. So in that way, yes, you could sort of have attentional or selective modulation based on different brain states or needs or demands of the animal at the time.
We've got one question here from Kara in the audience, which is, as I understand, you're mainly recording activity of odorants that are of similar chemical structure.
Yeah.
Have you observed antagonism to a similar level for innately relevant odorants within either the epithelium or in the olfactory bulb, the AOB?
Good question, right? These are all molecular, monomolecular odors, so it sort of occludes our interpretation in some ways, right? They're super pure and they don't arise in nature on their own. One way that I've sort of addressed that is by using a subset of odors that were molecularly distinct from the panel that we use. So these are all odors that typically have an ester group. So I used another panel that had non-esters and looked for antagonism between them. And we still found antagonism in most of the odor pairs that we chose. But to answer your question more directly about naturalistic odors, it's a tough question, right? Because any meaningful odor to the animal from like an ethological standpoint, like hormonal or social odors, they're actually blends of odors. They're not a single odor. So they're going to be activating or competing themselves for the receptors in the nose. So it's really hard to disentangle the effects of sort of self from those naturalistic signals compared to, say, another hormonal signal from a different animal. So that there's antagonism occurring on two different levels potentially in a system like that. But yeah, we've tried a little bit some experiments by collecting urines or other mouse smells to look at if there's some socially privileged odors and what the relationship to these nonlinearities is. And I'm finding it very difficult to collect concentrations which give reliable responses at the sensory neurons. So it's like the concentration of the odor is so low that we can't evoke responses that we're able to capture with the GCAMs that we have.
Great. All right, thanks for a great talk.
Can I ask one more question? So you have a very nice data set of this, basically the same experiments recorded at the nose and also the axon terminals, right? Does this, have you tried to see if there's any, what is added by presynaptic inhibition at the level of axons? Is there anything qualitatively different from interactions in the nose?
I've informally calculated this or looked at it. And I can tell you that there's, maybe about 10 to 15% more suppression at the level of the axon terminals. So work from John McGann and Matt Łakowiak says that this lateral inhibition in the glomerular layer shouldn't be mediated through the short axon cells, and it should only be mediated through the uniglomerular PG cells. Other people don't necessarily think that that's the case. So our work sort of suggests that that there probably is some lateral effects happening in the glomerular layer.
Yeah, very nice. Thank you very much for the excellent talk. Should we move on to the next talk? Okay, very great. So the second speaker today is Marino Pagan from Carlos Brody's lab at Princeton. So it looks like he's in the lab, and his animals will be trained while he gives his talk on individual variability of neural mechanisms underlying flexible decision making. So please take it on.
All right, thank you very much for the introduction. Let me see if I can share my screen. All right. I hope, can you hear me okay? Yes? Okay, I hope so. All right, so
yes, we can hear you well. Yeah.
Wonderful, thank you very much. Okay, so thank you very much for the introduction. And first of all, I would like to thank the organizers for the opportunity to speak today. The work I will present is about the neural mechanisms underlying our ability to flexibly select the relevant information to guide our decisions. For example, imagine being at a crowded meeting. Hopefully we will have one of those again soon, and you hear your name called out. If you want to locate where the person calling your name is, you will focus on the location of the sound and respond accordingly. But if you want to figure out who the person calling you is, then you might pay attention instead to the frequency or the pitch of the sound and respond according to that. And this ability to flexibly shift our focus to different features of a noisy stimulus is a fundamental cognitive process, but the underlying neural mechanisms are still largely unknown. To address this issue, I trained rats to perform a task requiring context-dependent selection and integration of sensory information, inspired by recent influential work in macaque monkeys. So at the beginning of each trial, rats are cued to focus on one of two possible stimulus features, location or frequency. After the cue, the rat is presented with a train of randomly timed auditory pulses, where each pulse is played either from the left or from the right, and each pulse is either high frequency, like beep, or low frequency, like boop. Now, in the location context, rats are rewarded if they turn at the end of the stimulus towards the side that had played the greater total number of pulses while ignoring the frequency of the pulses. And in contrast, in the frequency context, rats must ignore the location of the pulses, and they must turn left if the total number of low-frequency pulses was greater than the total number of high-frequency pulses, and turn right otherwise. And so because the two stimulus features are independent, an identical stimulus can be associated with opposite correct responses in the two contexts. The strength of the frequency evidence can be parametrically titrated by changing the average rates of high frequency versus low frequency pulses. So you can have a trial with strong prevalent evidence for low or strong prevalence evidence for high. And then independently, I can also vary the strength of the location evidence by varying whether the pulses are presented mostly on the left or on the right. And across different trials, I can use different rates so that I can tile this whole plane. When I first designed this task, I wasn't sure whether rats could actually learn it, especially because it's pretty challenging even for monkeys to learn this kind of behavior. So I was very excited to find out that rats can learn this task. And in fact, they can be trained to mastery with an automated procedure in a matter of a few months. Here I'm plotting the psychometric curves for 10 rats, and in blacks are shown here the curves for trials where the rats were cued to determine the prevalent location of the pulses. And as you can see, the choices reflect strongly the location of the pulses, but not their frequency. On the other hand, on trials where the rats were cued to determine the prevalent frequency of the pulses, here in blue, the choices reflect the prevalent frequency, but not the location of the pulses. So overall, this approach gives me two key advantages. First, by providing evidence in highly random but precisely known pulses, I have high statistical power to precisely characterize the animal's behavior and neural responses. And second, because I'm using rodents and an automated training procedure, I can study the individual variability across many trained subjects. So how does the brain perform this computation? Two major classes of models have been proposed. The first model proposes that sensory representations are modulated in a top-down fashion according to context to enhance relevant information and filter out irrelevant stimuli, and that only relevant information is passed in a feed-forward manner onto frontal areas to form decisions. So when we're in the frequency context, frequency information is passed to frontal regions, and when we're in the location context, location information is now selected. A second alternative model proposes instead that all information, relevant and irrelevant, is passed onto frontal regions, where recurrent computations operate to simultaneously select and integrate relevant inputs. So for example, in the frequency context, the recurrent dynamics will select and integrate frequency information, whereas in the location context, the recurrent circuit will select and integrate location information. And this second model was proposed in a recent influential study by Valerio Mante and David Susilo, working with Krishna Shenoy and Bill Newsom, where they found that contextually irrelevant evidence was still represented in frontal premotor regions in the monkey, such as the macaque frontal eye fields, or FEF. So for the rest of my talk, I will focus on data from a region in the rat brain called frontal orienting fields, or FOF, which is thought to be highly similar to primate FEF. And this is a frontal premotor region for the orienting movements that are used by the rat to express their choices. So this area is in a particularly good position to play a central role in this task, since it receives sensory auditory input, and it is also very close to the final motor output in this task. So using tetrads, I recorded the responses of single units in FOF in 4 rats. And here I'm showing the response of an example single neuron in FOF, where I'm plotting the average response as a function of time for trials where the rat chose left, here in black, or trials where the rat chose right, here in red. And this shows the classic ramping response that diverges for the two choices when the stimulus starts. Now, these solid lines were for the location context, but the response for this neuron is also very similar in the frequency context, here shown with dotted lines. So for this particular neurons, it mostly encodes the upcoming choice independently from the context. However, unlike this particular cell, most of the neurons in FOF are highly heterogeneous, and their responses encode a mixture of several task variables, such as choice, context, or the strength of the sensory stimulus. And this high degree of heterogeneity and mixed selectivity really makes it difficult to interpret the responses of individual neurons. So it's much more useful to study these neural responses at the level of the population instead. So what this means is that I can think of these individual responses for a given trial as a single trajectory in a high-dimensional rate space, where each axis represents the firing rate of one neuron. I can then take this high-dimensional population dynamics and project them onto a choice axis, which is the direction in neural space that is maximally predictive of the future decision, as well as feature axis that best represent the momentary strength of evidence for the two features. So now let me show you what these population dynamics in FOF look like when I average them across trials with the same context and same stimulus strength. So if we are in the location context here, on trials with strong location evidence to the right, meaning almost all pulses came from the right, the population response marches forward onto the choice axis. Whereas on trials with strong location evidence to the left, the trajectory moves to the left. So this movement onto the choice axis is really a reflection of the accumulation of evidence towards a decision. But on top of that, the population dynamics also form these arcs. And this modulation along the feature dimension is really a reflection of the strength of the momentary sensory evidence. Now, if you look at the average response for different strengths of frequency evidence in the frequency context, I get a very similar picture. And now what's really interesting is that the signal representing this signal representing the momentary sensory evidence is present also when that sensory signal is irrelevant. So I find a strong deflection according to the strength of frequency evidence during the location context, and a strong deflection according to the strength of the location evidence during the frequency context. And what this means is that the context-irrelevant information is not gated before reaching FOF and is still represented in this area. And these population dynamics that I observe in rat FOF are strikingly similar to those previously reported by Mante and Susilo in area FEF of monkeys performing an extremely similar task. So what models are compatible with these population dynamics? And from this point on, I will always plot relevant evidence in green and irrelevant in purple. So first, let's consider an extreme form of the first model that I presented before, where all the gating is performed in a feed-forward manner by top-down contextual modulation of sensory inputs, so that only relevant evidence is passed onto frontal regions to form a decision. In this case, neural trajectories would form almost straight lines onto the choice axis with no deflection onto the sensory axis. So relevant information will be collinear to the choice information, and irrelevant information does not percolate to frontal cortex, and it has overall a negligible impact on the population responses. And as you can see, this model is really incompatible with the data that we observe in frontal cortex in the rat or the monkey. In contrast, in model 2, both relevant and irrelevant information reaches frontal cortex, so the prediction is that there is a significant modulation along the feature axis, similar to the data in frontal cortex. And what I'm showing you here are the results for this analysis applied to neural networks that are constructed to behave as model 1 or as model 2. So at first, this first model seems incompatible with the neural data, but we can imagine a slightly different version of this model where computation is still performed in the same way with this top-down modulation and then this feed-forward projection of the relevant signal that is then used to form the decision. But now we're moving to the frontal cortex as a number of neurons that encode unfiltered sensory information, but that are totally disconnected from the rest of the circuit and that are not at all involved in how the computation is carried out. In this case, there will still be now relevant information that is not collinear with the choice. So you will see modulation here. And in addition, irrelevant sensory evidence will now modulate frontal population responses. So now also for module 1, you get traces that are quite similar to the neural data. And it's much more difficult to decide whether the data looks more like model 1 or model 2. A more fundamental difference between these two models is that model 1 is fast, meaning that if you're told that this is a location trial where location is the relevant feature, you can set those pathways up even before any sensory information has arrived. So as soon as the sensory information arrives, it is immediately selected. Whereas model 2 is slow because the relevant feature is selected only through ongoing frontal cortex recurrent dynamics that start only after the arrival of the sensory information. So what this means is if I analyze instead the responses to single instantaneous pulses of evidence, then I have a chance to distinguish between these two models. Because in the first model, a relevant pulse of evidence will be immediately selected to cause a step forward onto the potent choice axis, whereas an irrelevant pulse, here in purple, will induce dynamics that are confined entirely in the feature axis, which don't influence the decision. On the other hand, in the case of the second model, there's no immediate gating of the irrelevant evidence, meaning that both a pulse of relevant and a pulse of irrelevant evidence will cause similar initial deviation onto the choice axis. And in this case, gating depends on slower, context-dependent recurrent dynamics, so that a relevant pulse relaxes forward on the choice axis, while an irrelevant pulse relaxes back to the starting point. And it's also worth noting that these two selection mechanisms can coexist. And what we're really asking is where we lie on the spectrum, between these two models. And we can simply look, the simplest index you can think of is just looking at the separation of the initial responses on the choice axis between relevant and irrelevant to quantify the position on the spectrum, with model 1 predicting large separation and model 2 predicting a small separation. So let me take you through how I estimate single pulse responses from the data. As I mentioned earlier, it is crucial that in my task, evidence is presented in random but precisely known pulses. Now, on any given trial, I can consider what was the presented location evidence and whether it was presented in the location context or in the frequency context. Similarly, frequency evidence could be presented in the location context or in the frequency context. And I'm going to treat these 4 signals all in different ways. My goal is really to build a model of each neuron's firing rate, and to do so by characterizing the impact of each individual pulse on the neuron's firing rate as a temporal trace that is time-locked to when the pulse occurred. And different pulse types will produce different traces. So we'll have one trace for location evidence in the location context, one for location evidence in the frequency context, one for frequency evidence in the frequency context, and one for frequency evidence in the location context. In addition, I will have one component that doesn't depend on the pulses at all, but just depends on the time, another based on the current context, and another one based on the final choice of the animal. And my goal is to find the best set of temporal kernels to match the firing rate of the neuron across many trials with many different pulse presentations. And the prediction of the model is given by the addition of all these terms. And when we compare it with the neural data, we get very good matches. So this is a good model for what each neuron looks like. Now, these kernels will now be very good estimates of what the neuron response is to a single pulse of evidence for these 4 different pulse types. And with those, I can now directly look at the pulse-evoked population responses. So here I will be plotting the pulse-evoked population response for a particular rat and for pulses of location evidence. Now here in green is the estimated population response to a single pulse of location evidence when it's relevant, meaning in the location context. And in purple is the response to the same pulse of location evidence, but in the irrelevant frequency context. And as you can see in this particular case, the initial response to relevant and irrelevant pulses is very similar, but the subsequent relaxation dynamics differ. So this type of dynamics is very similar to the second recurrent model that I presented before. However, if I look at the dynamics evoked by pulses of frequency evidence for the same rat, I see something different, since now the initial response is different in the 2 contexts with an irrelevant...
Sorry to interrupt. Is it just me, or did we lose your audio?
Can you not hear me? Hello?
Can
I
everyone
can
else
hear
hear him?
everything.
Okay,
Good.
so it's my
As far as I
technical
know.
problem. So
Yeah.
please keep going. Sorry.
Okay, yeah. Okay, can you hear me now?
Yeah, it's all good.
Okay, great. So, all right. So I presented what the population response for this, the pulse-evoked population response for this first rat looks like for pulses of location evidence. And as I said, this is similar to the second model. However, when I look at the dynamics evolved now by pulses of frequency evidence, so this is the same rat, but here I was looking at pulses of location evidence, and here's the response to pulses of frequency evidence. I see something very different because now the initial response is different in the 2 contexts, with an irrelevant pulse producing a response orthogonal to the choice axis, and the relevant instead having a significant component along the choice axis. And this dynamic with this large initial separation are instead reminiscent of the first model that I presented before. So the first surprising result is that even for the same rat, the 2 types of pulses, location and frequency, can produce very different neural trajectories. And this suggests that they're selected and accumulated using 2 different mechanisms. And second, different rats display a dazzling degree of individual variability in their pulse dynamics. In particular, the difference in the initial response to relevant and irrelevant pulses, here shown by these arrows, goes from very large to very small, suggesting that there's a continuum in terms of how much each animal mixes mechanisms corresponding to model 1 and model 2. And note that this variability is not simply due to noise, because if I split the data in half and recompute these curves, I obtain very similar results. Okay, so this was all based on the neural data, but are these effects, these neural differences, also reflected somewhere in the behavior? So let me start again from the cartoon of the pulse responses for the 2 models. And here what I really want to focus on is the projection onto the choice axis as a function of time. In the first model, a relevant pulse causes a step on the choice axis, whereas an irrelevant pulse causes no motion. And what this means is that each pulse is immediately selected and integrated. And therefore, the impact on behavior of a pulse occurring early or late in the pulse train is identical. So even a pulse occurring immediately before the end of the stimulus will be fully integrated to impact the rat's choices, to impact choice if it's relevant, or it will be completely disregarded if it's irrelevant. And all pulses will be weighed approximately equally throughout stimulus presentation. So all of the so this is a plot of how much pulses impact choice as a function of time within the pulse train, with all the relevant pulses having a large constant weight and all the irrelevant pulses having a constant, very small weight. On the other hand, in the second model, both relevant and irrelevant pulses cause an initial small and identical deflection that is then amplified over time in the case of a relevant pulse and is suppressed over time in case of an irrelevant pulse. And so in this case, the prediction is that the early pulses will have enough time to get fully selected and integrated, but late pulses will not. And for late pulses, relevant or irrelevant pulses will have a similar impact on the choices. So the prediction is that when there's a big initial difference in the neural responses, the late pulses are correctly selected and integrated. But when there's no initial separation in the neural responses, this context-dependent modulation of the late pulses is not correctly performed. And this is exactly what I see in my data. So here I'm showing again the neural data, where I'm showing here the weights of so this is the neural data, and these now are the weight on the behavioral choices of relevant and irrelevant pulses presented at different time within the pulse train. And this is computed using a logistic regression. So this is analysis that only used the behavioral choices of the rats and not the neural data at all. So for my first rat, when there was a small where there was a small initial difference in the neural responses to relevant or irrelevant pulses of location evidence, I also found that there is a small difference in the impact on choices of late pulses of location evidence, whether they are relevant or irrelevant. But for pulses of frequency evidence, where there is a large initial neural difference, I also find a large behavioral differences in the late pulses. Now, this is just one rat, but this relationship holds well across all the rats that are recorded from. So if I plot these differences one against the other, you can see a strong positive correlation in my rat data. And here, that is here shown with these red circles. Between here, this is the neural index, and this is the behavioral index. So once again, what I find is that the behavioral weight on late pulses is well predicted by the shape of the neural dynamics. And this works also when I compare behavioral data and neural data from separate sessions. And this strongly suggests that these are both manifestations of the underlying neural mechanisms that are used to perform the computation, and that this mechanism is highly variable across different rats. Also, I don't have time to go into a lot of detail, but I just want to mention that I observed the same exact phenomenon across many different recurrent neural networks trained to perform this task. And when applying the same analysis to trained RNNs, I find the same positive correlation between these two indices, with this large degree also of variability. So my first conclusion is that individuals can use different algorithms to achieve the same computation. So here the question turned out not to really be which one is overall correct, model 1 or model 2, because they're both correct. And it's really more about which combination any given animal is using for any given feature. Second, this issue of individual variability is particularly important in cognition, where we study algorithms that are completely internal or latent. So you could imagine having the same exact sensory stimuli and the same exact motor output, but different internal algorithms that connect the two. And it's really critical to identify what these algorithms are in different individuals. Another crucial point is that this is a problem because very often findings are reported only when they're consistent across animals. And this can obscure a lot of very important and very meaningful real biology. So we really need ways to measure and characterize each individual's computations. And the idea in this work is to study impulse responses for system identification, which is really an old idea that is widely used in engineering and many other fields, and that is potentially extremely helpful also in biology. And finally, my next steps will be to record also in sensory cortex, as well in many other areas. And really the ultimate goal is to understand the brain-wide networks that support flexible feature selection. I can now train many animals, so I really plan to try to record from as many brain areas that are positively involved in this kind of behavior as possible. So with that, I would like to thank Carlos, Mikio Aoi, who was a postdoc at Princeton with Jonathan Pillow, and that is now starting his own lab at UCSD, which was crucial to develop the analysis to compare rat and monkey data, and also in particular, the pulse-based analysis of neural responses. Valerio Mante and David Susilla that provide invaluable help about training RNNs, the comparison with the monkey experiment, and generally how to think about this problem. And Vincent, who helped collect some of the data, as well as all the other members of the Brody Lab. And also a big thank you to the Simons Foundation for the very generous support, as well as HHMI and NIH. And also thank you very much for your attention.
Well, that was a fantastic talk, Marino. And yeah, please everyone send your questions to your chat while I ask the first question. So yeah, I really like, so when you were presenting the two models at the beginning, my initial reaction was that, both must be possible, and it really depends on how each rat decides to solve this task. And then towards the end, you were showing that it is indeed the case, and it is variable from individual to individual. So I thought that was really nice. Do you have any sense of the origin of these individual variability? How stable are the strategies, at least behavioral strategies, within each animal? And is there anything you can do to predict the strategy of each animal? I might have said mouse, but I think you're using rats. Yeah.
Yeah, in rats. But this is, I think it's a really, really important and interesting question. It's something that I'm extremely interested in. So they're definitely stable insofar as both the neural data, if I just kind of look at a bunch of sessions recorded at a certain point, and then another a set of sessions recorded later on, I tend to see the similar indices. but also the behavior where I have a lot more data and a lot more animals, these are very, very stable. So if I look at those logistic regression, the weight of the relevant and irrelevant evidence as a function of time, those are pretty stable. And so what I'm really curious is like, where does the variability comes from? And I think one thing that I'm going to be focusing on is the training takes a pretty long time, takes many months. And so I really want to study how this behavior is shaped throughout training, and then eventually also record throughout training and see where different individuals might diverge and end up with different solutions. So I don't have an answer to that yet, but it's something that I'm extremely interested in to research in the future.
Yeah, very nice. Thank you.
All right, great talk. We have some questions from the audience. So Chuck Yin asks, have you found any neurons in FOF only encoding auditory stimulus information?
I think if I were to really try to look hard, I think I might. It's very extremely, if any, very, very few, and I would really need to. I think by far the strongest signal that you find in FOF is the choice signal. Is this kind of motion onto the choice axis that diverges for the two choice that the animal will make. That is by far the strongest signal. I do find whenever I see sensory information is generally also modulated by the context. So if I might be able to kind of isolate maybe one or two cells that I can say these are mostly sensory and don't see much contextual modulation. But it's I know that that's what I presented in the cartoon, but that's kind of just to say there can be sensory signals kind of riding on top. So that does not need to be actually implemented in individual neurons, because the individual neurons are extremely heterogeneous and just present all these mixtures. But the point is that there could be sensory information there that is not participating to the computation, but is just available in the area.
And to follow up on that, if you look at activity along the choice axis at the population level for single trials, I don't know if you have yields that are high enough to look at this, but can you actually detect either the animals changing their mind, or can you predict failures on a trial by trial basis?
I've started looking at it. I have a few sessions in which I have a decent number of simultaneously recorded neurons. It's definitely the case that the movement on the choice axis traces the choices that the animal makes. So on error trials, the movement on the choice axis traces the choice the animal makes and not what the correct choice was. I think I could see something. It's something that I plan to do next. I've not yet gone into too much detail with that analysis. So I can't. I think so, but I've not yet quantified that well in terms of like kind of changes of mind, and something that I know a lot of other labs have tested before. I'm pretty confident that that's the case. But yeah,
or prediction of reaction time.
Or prediction of in this case. So this is, but I must point out, this is not strictly speaking a reaction time task insofar as the animals must listen to the whole duration of the stimulus, which is 1.3 seconds, and only then is allowed to respond. However, there could be a correlation with how fast the moment the animal is allowed to respond will actually do the decision, depending on what I see on the movement on the choice axis. Yes.
Sure. All right, we've got a question from Suchitana Beas-Dutta, who says, you mentioned that there's variability among animals which would signify individuality and preference. Do you see a particular animal choosing preferred frequency or direction over time? Is this preference dependent on the internal state of the animal, and do you think it might be heritable?
So there's a few things there. So one, I'm not sure I interpret that correctly, but it seems to suggest by an animal preferring a frequency or a location, I imagine some kind of bias or like, an animal. And that is something that I work really hard to prevent, because my goal is for the animals to actually perform the task and not just always go right or always go towards the association with the, with the high frequency. So I have always a mechanism in place throughout the training and also throughout the behavior that whenever I recognize that the animal is starting to be biased, I start present more and more trials that kind of go against that bias. So if the animal tends to go more and more often to the right, I will present more and more trials where the correct answer is to the left to kind of try to counter that. Then one question was whether that is inheritable, and I think there could be genetic differences in at least in the ability of different strains to learn this kind of behavior, because this is again really hard, and I definitely do see differences. Some rats get it relatively quickly. For some, it's a much longer process. Some never get it. And I believe there might be a genetic component to that. And that's something, again, that I'm very interested in studying, but it's kind of a future direction. There might have been something else in that question that I am forgetting right now.
I think heritability was the last thing. Dependence on internal state. So I guess my interpretation of that is, do you see some kind of neural bias towards one or the other in individuals?
So I guess all I can say is that there are rats that find the location component of the task easier than the frequency, or they find in general, they tend to find the frequency component slightly easier. And I tend to see those reflected in the neural separation, right? So if there's a clear separation in the neural responses for the relevant and irrelevant for the frequency that is large, then generally they also tend to be better at the behavior. So again, there's this kind of correlation between the behavioral measure of how well they can do the context-dependent selection and the neural one. But this is more kind of the selection part, which is part of the task, more than kind of preferring one over the other.
All right, one final question here. Pooja Ravishankar asks, are these directions dependent on the strategies dependent on the order in which they are trained, location or frequency first?
No, they're not, because all the animals go through exactly the same automated training procedure, and just arbitrarily, I always first train them on the location task. And the training, when I train the two tasks in isolation, is actually very fast. So I first train them on the location task alone, then I train them on the frequency task alone, and that they can get it very, very quickly. And then I start alternating the two, and that also they get very quickly. And then I start making it so that the same trial has both location and frequency information, and they have to pay attention to the context to know which one to pay attention to. And that's what's really hard, and that's what takes very long time. So again, these are all trained the same way, and so the fact that they come up with different answer tells me that it's not due to the training. And also, yeah, I don't think that part of the training, they go really fast. It's really the whole context-dependent selection that is the really hard part.
All right. Do we have time for more? Takaki, you mentioned you have to run shortly, I think.
Yeah, I do, but you guys can keep going.
All right.
We're about close to time, but if you have another question you wanted to add, there's no like hard deadline or anything. In the worst case, Takaki can dip out, and but we thank you before you leave. So go ahead if you have another question.
I've got sort of a technical question. So I assume you're fitting one of the Park Pillow type GLMs for single neuron responses, which is great because the frequency of the pulse trains is high enough they'll be overlapping. So that makes sense. How do you go from that then to the neural population trajectories?
So I can maybe pull up the slide again. So the idea is so this is the slide for that. So I fit this different kernels, right, for each neuron. So for each neuron, I end up with these temporal kernels that are aligned to the pulse presentations for these 4 pulse types. And then all I'm just doing is then I'm treating these kernels as the response of that neuron to that particular pulse type. And so what I do is then I put multiple kernels together for all the neurons in my population, and that is what I'm treating as the pulse-evoked population response. In fact, so this is the kernel that comes out of the GLM, but I have also tested using recurrent neural networks that the results of the GLM is actually a very good approximation of what the actual response to a single pulse is. I can pull that up. So here is in recurrent neural networks, I have perfect information about how the network responds to individual pulses. So I can either present the trials like in my task with trains of pulses, and then apply the GLM to retrieve these kernels, Or I can present a single pulse and then see what the response is like. And I can find that the response to a single pulse is actually very similar to the estimate of the GLM. So what I do next is I look at these pulses for different neurons and combine them into the population response. And so I hope that.
But then to get the choice dimension, you're doing LDA for left versus right or something like that?
Yeah, the choice axis is essentially the first PCA axis for the choice kernel. And another thing I can mention is that it's very one-dimensional. It's not something that I'm forcing onto the data, the idea of just having a single choice axis, because if I really look at the responses, just depending on the choice, they really kind of move onto a single dimension. Like it captures 80-90% of the variance. If I just look at them based on the choice and not the other aspects of the task.
Yeah, I really like this pulse-aligned analysis. I think that's a powerful way to think about this. All right, so I guess we're a little over time here. Should we go ahead and wrap up?
Yeah, I think that was awesome. Thanks to Joe and Marino for that. We've gone over time, so we'll end it here. We hope that everybody can come for the next one. You can find everything in our website, and if you want to sign up for some updates on the weekly talks, click the link that we posted right here in the chat. And pretty soon after this season's over, we'll open up another season where we'll call out for abstracts, and we'll hopefully grow this even more so than now. But thanks for 2 excellent talks, and thanks Britton and Takaki, who's gone now, for moderating. That was really cool. That was just 2 fantastic talks. So we'll see you next time. Thanks a lot for coming.
Thanks a lot, everyone.